Hello and welcome to our webinar on CSF1, CSF1R and macrophages. I'm Oliver Rosenwasser and I'm the product manager for the veterinary antibody products at Bio-Rad. I will introduce you to our speaker in a moment, but would first like to let you know that this webinar will be recorded and an on-demand version will be available soon on the Bio-Rad website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide which gives solutions for the most common technical problems. We would like to make this webinar as interactive as possible, so please submit questions or comments via the Q&A box located at the bottom of the screen. You can do this throughout the webinar. We will answer as many questions as possible at the end of the presentation. Should any questions remain unanswered, we will email you a reply. And now, I would like to introduce Professor David Hume. Professor David Hume is the Director of the Roslin Institute at the University of Edinburgh and Research Director at the Royal School of Veterinary Studies. His previous role was Director of the ARC Special Research Centre for Functional and Applied Genomics at the Institute for Molecular Biosciences at the University of Queensland. Professor Hume is an international authority in genome sciences with a particular focus on the function of macrophages and their role in infection, inflammatory disease and cancer. His current research exploits the opportunities of the Roslin Institute, a multidisciplinary animal sciences institute, to explore comparative aspects of macrophage differentiation, function and the roles various factors have on that process. This webinar covers some aspects of this work. And now over to you, David.
Thanks, Oliver. So, as Oliver said, I'm going to talk today about CSF1, otherwise known as macrophage colony stimulating factor, and its receptor, the CSF1 receptor, and their role in macrophage differentiation. So why do we want to study macrophages? Macrophages really are the classic double-edged sword of the immune system. On the one hand, they're absolutely required for normal development, wound repair, and particularly for defense against pathogens. And on the other hand, macrophage products mediate much of the pathology of many of the diseases shown in this slide. The consequence of their role in the immune system is that their function is under intense immune selection. And so there's a great deal of variation in evolutionary terms in the genes expressed in macrophages, both within and between species. The image in this picture shows a macrophage migrating from left to right, and it has been immunostained in yellow with to detect the intracellular protein TNF-alpha, and this cell is actually going to secrete this potentially toxic molecule to the end of the processes that it's extending from left to right. We traditionally view macrophages as inflammatory cells, but there are actually large numbers of macrophages in every organ in the body. I started my career in macrophage biology locating macrophages with the monoclonal antibody F480 in tissues of the mouse. And this image shows macrophages in the kidney at top left, the wall of the gut, the retina where we can see microglia, the bone marrow cavity, and at the bottom we can see macrophage migrating towards a dying cell, the small blue nucleus that you can see in the picture, which is going to be engulfed by the migrating macrophage. So resident macrophages are extremely important for homeostasis. The macrophages in every tissue of the body, as well as the inflammatory macrophages, form part of a family of cells, starting with progenitors in the bone marrow, the hematopoietic stem cell, the common myeloid progenitor, the granulocyte macrophage progenitor, and the promonocyte, which migrate into the blood to form blood monocytes, and then migrate into the tissues to give rise to the tissue macrophage populations, some of which are also capable of local self-replication. The control of the proliferation and differentiation of all of the members of this family, the mononuclear phagocyte system, is controlled by macrophage colony stimulating factor, or CSF1. CSF1 mediates its actions by binding to a cell surface receptor, the CSF1 receptor, and the picture on the left of this image shows the structure of the CSF1 receptor, which has 5 extracellular immunoglobulin-like domains and an intracellular tyrosine kinase domain. The activation of the receptor involves dimerization and autophosphorylation, which then links phosphorylated tyrosine to a downstream signaling pathway. There are really 4 major functions of the receptor and the signaling. It signals for proliferation and differentiation. It's also required for survival of mature macrophages, which will die if you block the receptor. The receptor mediates endocytosis and removes the ligand from the circulation. And then there is also acute regulation of particular functions of the macrophages that bear the reception. We know about the function of the receptor for CSF1 because we have animals that lack either the ligand or the receptor through a natural mutation in both mouse and rat, or targeted mutagenesis of the receptor. And those animals are severely compromised in their development. They're very small at birth, they fail to thrive, they have severe bone phenotypes, and they have defects in development of all sorts of organ systems. The receptor knockout in particular in mice is severely compromised and seldom lives for more than 3 weeks postnatally. So the receptor for CSF1, not surprisingly because the biology is macrophage focused, is expressed only in cells of the macrophage lineage. And some years ago my group decided to use this fact to create a macrophage-specific transgenic reporter, and what we did was identify the regions of the CSF1 receptor gene that are conserved across species, and we extracted a segment of mouse genomic DNA and placed it upstream of the enhanced green fluorescent protein reporter to make a CSF1 receptor EGFP reporter gene. That work was originally published in Blood in 2003. This approach to making a reporter gene worked rather wondrously. We generated mice that we refer to as the MacGreen mice, which have been distributed all around the world and are available from the Jackson laboratories now. And the first observation that we made with these mice is that all of the macrophages that we could isolate from the peritoneal cavity, the thioglycolate elicited peritoneal macrophages, or TEPM, that we could grow from the bone marrow, BMMs, or that we could lavage from the bronchoalveolar space, so-called bronchoalveolar lavage macrophages or alveolar macrophages, were all highly positive for the transgene. If we look at the green fluorescent profiles in the middle of this picture, we see the peaks of positive cells in all 3 of these populations. We also showed that the transgene had to include the first intron, in particular a very conserved element that we call the FIMS intronic regulatory element, or FIRE. And if we deleted that element, as you can see, or we lost the whole first intron, in the red panels you can see there's no expression in macrophages, and if we just take out that highly conserved 300 base pair element, the expression disappears altogether. So when we looked at the tissues of the MacGreen mice, we realized that we had stumbled on something that was even better than we had hoped, even better than the immunohistochemistry that we could do with F480 or other antibodies. This fluorescent marker is expressed in all cells of the mononuclear phagocyte lineage, and we can see the large numbers of them in the brain, in the retina. These are so-called microglia, in the kidney, the interstitial cells in the testis, the osteoclasts on the surface of bone, the so-called Langerhans cells and dermal macrophages in the skin, which we can see in the ear, and which we can also see in a flat mount of epidermis, and the large numbers of macrophages in the thymus. And many of these macrophages have now been isolated and profiled, and we start to learn about the way in which their function differs between the different organ systems. With improvements in imaging technologies, particularly with two-photon microscopy, which can give much greater resolution, we have used the macrine transgene to look at macrophages in detail in unprecedented resolution. And this particular image shows the Kupffer cells, the macrophages of the liver, with the macrine transgene. And you can see the way that these macrophages extend processes into the surrounding tissue. And in fact, every hepatocyte is in intimate contact with macrophage processes. So the presence of the CSF1 receptor on every macrophage and its potential role in the biology of macrophages encouraged us to ask, what happens if we block the function of that receptor? As I said earlier, we knew that natural knockouts or induced knockouts of either the ligand or the receptor have profound effects, but that's rather different from blocking its function in an adult animal as a prelude to potentially using the antibody therapeutically. To introduce the effect of CSF1 anti-CSF1 receptor antibodies, I need to introduce the concept of subpopulations. In both mice and humans, the peripheral blood monocytes, or the macrophages of the blood, can be divided into at least two functional subpopulations. One population which expresses the chemokine receptor CX3CR1, and this is actually an error on the slide, low levels of the LPS receptor CD14, is the precursor of most resident tissue macrophages, and a subset of these cells actually patrol the vessel wall. A second population which expresses low levels of the fractalkine or CX3CR1 receptor and high levels in mice of the marker Ly6C and in humans CD14, these cells, which are referred to as classical monocytes, have a short half-life, and they are the immediate precursors of inflammatory macrophages. So the CSF1 receptor varies between these populations, and one of the first questions we wanted to ask with an antibody against the receptor is what happens to monocyte differentiation. So this slide shows the profiles that you can see in the publication from another research group in 2010 who characterized and compared the monocytes from humans and mice. The top three panels show the profile of expression of CD14 and the Fc receptor CD16 on human peripheral blood monocytes. And what you can see is that there is, in the right-hand panel, really a crescent and continuous profile of CD14, and whereas CD16 is restricted to a subpopulation of cells that have low CD14. In the bottom, we look at the profiles of mouse peripheral blood monocytes, and in the right side panel, we see the staining for the Ly6C or GR1 marker against CD115, which is the CSF1 receptor. And you can see that all of these cells are in fact CSF1 receptor positive. It's not so obvious on this slide, but there is in fact a gradation of the level of CD115 between the GR1 high and GR1 low cells. So this slide then shows what happens when you treat a mouse with a blocking antibody against the CSF1 receptor. And what we're looking at here is using the macrine transgene as a reporter, comparing double staining for the transgene and expression of the macrophage marker F480 or the subset specific marker GR1 or Ly6C. What you can see in the upper panel, we can identify two populations of F480 positive cells. They differ in the absolute level of expression of F480, and the F480 high cells have somewhat higher expression of the transgene. And on the right you can see that when we treat with antibody against the CSF1 receptor, we specifically lose that population in the R3 circle. If we look in the bottom panel, now looking at GR1, you can see there is a GR1 low population that is high for the transgene, the R4 box, and you can see that the cells in that box have been completely ablated. The panels at right actually show, which I won't go through in detail, M279 is the anti-CSF1 receptor antibody which was made by Amgen, collaborators in this project. And what you can see is that the total number of blood monocytes actually doesn't change. So what happens with the antibody is that the cells collapse down into the Ly6C high population. So as I said, what the antibody does is block monocyte maturation, differentiation. The antibody actually tells us that the Ly6C low or non-classical monocytes derive from the Ly6C high and are dependent on CSF1. So if we look at the tissues of the MacGreen mouse treated with antibody against the receptor, we very rapidly lose most of the tissue macrophage populations. In particular, the macrophages of the liver, which we can see in this image. At left we see the macrophages all over the liver, bright green with the transgene, and at right we see a liver that is almost devoid of resident macrophages or Kupffer cells. Similarly, and rather spectacularly, the macrophage population of the wall of the gut, which fills the lamina propria and also the muscularis, has substantial numbers of these cells. As you can see at right, the macrophage population is almost completely ablated by the treatment with anti-CSF1 receptor antibody. Of course, the reason that we worked on this antibody in part is to explore the possibility of using it therapeutically, and we tested it in a number of tumor models. The mouse is not a great model for tumor biology because of the relative size and requirement for vascularization. But what we could do is see whether we can prevent macrophage accumulation in a tumor. And as you can see, and this is a mesothelioma model, the transgene is a rather more spectacular way of seeing the macrophages in the tissues, but what you can see is that treatment with the anti-CSF1 receptor antibody as the tumor is growing almost completely prevented macrophage accumulation in the tumor. One of the particularly interesting areas for us to look at was the bone. It has been known for a long time that CSF1 is required for the generation of osteoclasts, which are the cells that resorb bone. It's not widely recognized that the C57 black 6 mouse in particular is a natural model of osteoporosis, and female C57 black 6 mice reduce their bone density quite rapidly with age. By 12 to 16 weeks, they have a dramatic reduction in bone density compared to male mice. And what you can see in the upper left, the actual graph, but it's rather spectacularly obvious in the micro CT pictures of bone that treatment with the anti-CSF1 receptor antibody can prevent age-dependent osteoporosis in the mouse model. The way that it does so, and it's not so clear on the images, is by drastically reducing the number of osteoclasts on the bone surface in the right-hand image. These are stained with for the activity of the enzyme TRAP, or tartrate-resistant acid phosphatase, and the osteoclasts are largely ablated by the antibody treatment. one of the unexpected phenotypes of depletion of macrophages that we observed was in the wall of the gut. As you can see, and I showed you earlier, the transgene detects massive numbers of macrophages within the lamina propria. In the bottom right image with the transgene, you can see the macrophages surrounding the crypts, which are where epithelial cell proliferation is concentrated. And within those crypts are cells called Paneth cells, which in the top left image are stained for the presence of the enzyme lysozyme. What you can see is that the antibody treatment completely ablates the Paneth cells, and it was actually known previously that CSF1 deficient mice are also deficient in Paneth cells. But what you can also see is that the receptor is not expressed at all in the Paneth cells or in any epithelial cells. The depletion of the lamina propria macrophages with the antibody also increases the number of mucus-secreting goblet cells, and essentially the overall view is that the macrophages surrounding the crypts actually control the differentiation of the epithelial cells. So to summarize what we've seen with an antibody against the CSF1 receptor, we deplete the resident monocytes or the mature non-classical monocytes in blood. We get selective depletion of resident macrophages, including cells that some would consider to be dendritic cells, in most organs of the body. The exceptions are the lung, the brain, the uterus, and parts of lymphoid tissues. We've actually tested this antibody in a number of models of inflammation, and it has no effect. What it does do is deplete macrophages from a range of tumors in different models, suggesting that when a tumor grows, essentially what it does is recruit resident-type macrophages. The one thing I haven't talked about is that if we pre-treat mice with the antibody and then do a bone marrow transplant, they become extremely sensitive to graft versus host disease. Graft versus host disease is a disease of the intestine, essentially, and what this suggests is that the resident macrophages of the intestinal wall are actually important in protecting against inflammation. So we have a new model using this antibody to study the function of macrophages in homeostasis and disease processes. So that deals with the effect of a blocking antibody of interfering with CSF1 receptor function, and there are many drug companies in the space of making receptor kinase inhibitors as well. What happens if we increase the number of macrophages and alter their gene expression with CSF1? Could CSF1 have a therapeutic potential? And we and others have published a number of papers suggesting that indeed CSF1 could be a treatment for a wide range of inflammatory diseases, and particularly to accelerate healing in damage situations and trauma. The problem with using CSF1 as a therapeutic agent is that the molecule is relatively small. The active component is only 150 amino acids, and if it's injected into the circulation, it's very rapidly cleared through renal filtration. CSF1 was actually tested for therapeutic potential when it was first cloned back in the 1980s, and it was biologically active by continuous infusion. But continuous infusion is really not a particularly practical therapeutic option. To get around this problem with collaboration with the animal health company Zoetis, we developed a immunoglobulin fusion protein, or FC fusion protein of CSF1, as shown in the upper portion of this slide. And we initially made the pig molecule because pig CSF1 is active in both humans and, in fact, every species that we've looked at, as well as rodents. And so what we showed, and I think the bottom two panels show this very clearly, is that if we look at CSF1 alone, recombinant pig CSF1 injected into a pig, it reaches a peak concentration quite quickly and then is essentially all gone within 24 hours. If we inject the Fc fusion protein into a pig, then the concentration is massively elevated. The peak concentration is almost an order of magnitude higher than with the native molecule, and the elevated level of circulating CSF1 is maintained for at least 72 hours. So we first asked what happens if you inject this Fc conjugate into mice, and not surprisingly, in peripheral blood you see a significant increase in the total white blood cell count, a massive increase in the number of circulating blood monocytes, smaller increase in the number of neutrophils. The reason for this is in large measure because CSF1 promotes the production of GCSF, the growth factor for granulocytes, indirectly because that is a product of macrophages. And there is also a much smaller change, a small reduction in the lymphocyte count. So one of the things that we didn't expect when we did this was that CSF1 treatment actually increases body weight by approximately a gram relative to the untreated or the PBS-treated controls. But this increase in body weight is almost entirely attributable to an increase in the weight of the spleen and the liver as a proportion of body weight. In fact, the treatment increased the weight of the liver by 30 to 40% relative to the controls. This is an unprecedented response. As we would expect, alongside the increase in the number of blood monocytes, we could detect using our transgene or using the F480 immunohistochemical marker at the bottom right, a very large increase in the numbers of macrophages in every organ that we looked in. We're showing here the lung, the spleen, and the liver. In the spleen, we're showing in the control injected, we can see clearly delineated the white pulp and the red pulp, whereas in the CSF1 treated spleens, there is large-scale infiltration of the white pulp by GFP-expressing macrophages, so that the delineation of red and white pulp is much more difficult to see. And the same you can see in the bottom right. When you look with F480, there's F480 positive cells infiltrating much of the white pulp. At least some of the effects that we see on the blood monocyte numbers and tissue macrophage numbers reflect a very large increase in the production of monocytes and macrophages from the bone marrow. We looked specifically in the bottom panel at the proportions of GFP positive cells, the transgene, the numbers of F480 positive macrophages and GR1 positive monocyte precursors, and you can see that in every case the treatment increased their numbers in the bone marrow. We also looked at the osteoclasts. So you recall that the antibody against the CSF1 receptor depleted the osteoclasts. On the other hand, the CSF1 treatment massively increased the numbers of osteoclasts on the bone surface. Surprisingly, that actually didn't produce any change in bone density, and so what it seems to do is increase the turnover in bone, and if anything, in longer-term treatment, it increases bone density. Although the macrophage content of both spleen and liver certainly increases, the level of increase is nothing like enough to explain the increase in size. The predominant differential is an increase, particularly in the liver, in the numbers and density of hepatocytes. And what we could see by staining for proliferating cell nuclear antigen, the brown stain, normally in an adult mouse there are very few proliferating hepatocytes, whereas in the CSF1 treated mice, there is a massive increase in the number of proliferating hepatocytes. We also see evidence of this in the spleen, particularly within the white pulp and within germinal centers. The numbers of proliferating cells were greatly increased by the treatment with CSF1. In order to work out exactly how this increase in hepatocyte proliferation occurs in response to CSF1 FC treatment, we expression profiled the livers of the treated and untreated animals alongside bone marrow-derived macrophages grown in CSF1 and treated with bacterial lipopolysaccharide. The reason we looked at lipopolysaccharide is that the liver is exposed to bacterial products coming in the portal vein, and we wish to ascertain whether the newly recruited macrophages might be responding to products coming in from the gut. I won't go through the details. Some of you may be able to read these results online. But the method that we've used here to look at the data is a proprietary software called BioLayout Express 3D. This software enables one to visualize transcriptional networks and the way that genes are co-expressed with each other. And the graphs at left show the clusters of co-expressed genes colored differently, and the graphs at right show the average profiles of those co-expressed genes. What we were able to see was that there was a set of genes that are highly inducible in macrophages by LPS that were also induced in the liver, and they include some of the inflammatory cytokines like IL-1, IL-6, and TNF, which are known to stimulate the proliferation of hepatocytes. We could also see the induction within the liver of many cell cycle related genes, which again confirmed to us that there was extensive hepatocyte proliferation going on. These studies obviously encouraged us to think about therapeutic applications of CSF1 FC, in particular in liver injury, and so we, in the paper published last year in Science Translational Medicine, we explored CSF1 FC treatment in a number of mouse models of liver injury. We were able to show in partial hepatectomy that CSF1 FC treatment accelerated the recovery of the size of the liver, and you can see in the bottom panels that it also increased what was already a significant increase in hepatocyte proliferation. Similarly, in a mouse model of acute liver failure caused by paracetamol, or in a model of chronic liver injury followed by partial hepatectomy, which is a fairly common human scenario. In each case, the treatment accelerated the recovery of liver function. So obviously these results are encouraging, but mice are not small humans. Their metabolism, their rates of growth, cell cycle, etc. are very different. And so we wanted to extend the results to a large animal that is more human-like, and we have just recently published in the American Journal of Physiology that the treatment has the same effect in pigs. The slides at the top show the effect of CSF1 treatment on proliferation in the liver of pigs. This is with staining with the Ki67 nuclear marker for proliferating cells. In the bottom part of the panel, you see the very large increase in the proportion of Ki67 positive cells in response to CSF1. And what you also see is that the nuclear density in any field is substantially increased by the CSF1 treatment, consistent with hyperplasia being the major driver of the growth of the liver. And so obviously we're very optimistic that CSF1 will find, or the FC fusion protein will find an application in human liver regeneration following injury, but this is the vanguard of applications in all forms of tissue repair. So CSF1 system is not unique to mammals. Some years ago now, we identified CSF1 in birds, and also, although I haven't mentioned it so far in this talk, there is a second ligand for the CSF1 receptor called interleukin-34, which is also conserved in birds. The actual combining site of the receptor, we've actually modeled the CSF1 molecule and its receptor from crystal structures. The overall structure of all of the molecules is conserved between mammals and birds, as is the enhancer that was the basis for our CSF1 receptor transgene in mice, the MacGreen transgene. So we have made recombinant CSF1, recombinant IL-34, antibodies against the ligand and the receptor, and we have also recently made the CSF1 Fc fusion protein in birds. As I said, we have made a transgene reporter. These birds we make available to the bioscience community. We've made both red and green macrophage-specific transgenes in this paper in development. Some of the images show the rather spectacular ways that they can be used to visualize macrophages, including the red image, which shows that there are very large numbers of macrophages in skeletal muscle, which is actually also true in mice and has largely been ignored in terms of muscle physiology. The right-hand panel actually also shows the co-location of our transgene with our new antibody against the chicken CSF1 receptor, which is a very useful model for studying the biology of macrophages in birds. As I mentioned, we've made the equivalent chicken CSF1 Fc fusion protein and tested its biological impact on the macrophages in the birds, the direct repeat, if you like, of the experiments that we did in mice. And what you can see in this is that the macrophage numbers are so great that we can actually see them in a whole mount of the organs of chickens. And if we treat with the CSF1, the impact is so substantial that we can actually see it without even making sections. And on the right you can see sections of various organs of control and CSF1 treated birds. So what this shows is firstly that we can use the CSF1 Fc in birds to expand the macrophage populations, and of course inter alia that the biology of CSF1 as a macrophage growth factor is conserved between mammals and birds. So I've recently written a review on the biology of CSF1. The overview is actually in the abstract showing on your screen. All of our data shows that recombinant CSF1, especially the Fc fusion protein, can expand macrophage numbers, alter their phenotypes, and on the other hand that antibodies against the receptor can change the numbers of macrophages in tissues. Both antibodies and the growth factor may have applications in different kinds of disease processes. We've produced the antibodies against the mouse, the pig, and the chicken CSF1 receptor, and also ligands that will work across all of these species and reporter transgenes with which we can assay the biological impacts. And so I think the future is bright for therapeutic applications of CSF1, and we have the tools that we have developed to really understand macrophage biology. So thank you for your attention. I'd just like to mention my organization, the Roslin Institute, is a world-leading animal sciences institute located at the Easterbush campus just south of Edinburgh, with beautiful views of the Pentland Hills on the left and a modern research building. We're building an innovation centre next door to us and a large animal research centre on this campus. This is leading environment for animal sciences research. I'd also like to mention my many colleagues who have contributed to the work that I've described today.
Thank you, David, for your interesting and very wide-ranging presentation on CSF1 and macrophage biology. For our audience, please enter your questions now in the Q&A box if you haven't already done so. Before we move on to the Q&A section, I would like to point out a few of the resources that Bio-Rad has to offer to support research to the veterinary immune system. I hope you've already noticed the box on the top right of your screen containing key product flyers. These introduce our antibodies sorted by species. For full up-to-date information on antibodies and technical support resources, please navigate to bio-rad-antibodies.com/vet. So essentially our key website where the veterinary antibodies are. You'll find several of the key antibodies mentioned here in our portfolio. Now over to the Q&A section. Thank you for the many questions that you have sent in. We'll pick a few to answer, but the remaining ones will be answered by email, so everybody will get their questions answered. David, first question. Would you expect the pig CSF1 to work in other species than mouse and pig? And would the protein be immunogenic?
So we have actually tested it in rats as well, and I mentioned briefly in the talk, we chose the pig protein because we knew that it would work across all mammalian species. The mouse CSF1 does not work on the human CSF1 receptor, and it also doesn't work on some of the other large animals, although it does work on mice. We've actually published comparative analysis of exactly why this is the case. The pig CSF1, we've treated mice for actually in separate experiments for up to 4 weeks, and we've not seen an immune response against it. This is actually quite irritating for one reason, because we want to make antibodies against CSF1, and it's actually very difficult to get good antibodies against CSF1 because it seems not to generate decent immune responses. So basically the protein that we have, which we have made available through Bio-Rad, will work in every species that we're aware of, every mammalian species, but not in birds.
Oh, okay. Thank you. Next one. Do you think CSF1 would be likely to have therapeutic potential in tissue immunization other than the liver?
We actually have tested it in quite a number of situations. What we currently believe is that CSF1 as the sole stimulus promotes macrophages that favor tissue repair. So we've tested it in published work. We've tested it in kidney repair and in non-conjoined fractures, and we have work coming up on a number of other tissue regeneration scenarios. We've also tested it, as I mentioned in passing, in pigs, and we're moving in other indications into the pig as a large animal model.
Mm-hmm. Okay, thank you. Something possibly a bit closely related. Would CSF1 treatment be likely to alter the acquired immune response?
That's a very interesting question, and we've not tested it so much. What we think is that CSF1 as the sole stimulus is probably immunosuppressive, and it probably mediates some of the immunosuppression of advanced cancer and advanced infectious disease. But that we're not certain whether that's true in other species other than mice, because the unique feature of the mouse is that the macrophages grown in CSF1 do not seem to express class 2 MHC, whereas in rats they do, and certainly in humans and pigs they do. So we're not. That's the one area where we really need to move from the mouse as a model and look at other species. It's quite possible that CSF1 will actually turn out to be an adjuvant.
Okay. It's a little bit more specific now on the final question we have time for today. Is CSF1 expressed in microglia?
So the receptor is certainly expressed by microglia. The CSF1 ligand is not in any species that has been looked at. It's actually expressed by neurons, and there's a very intimate relationship between neurons and microglia. So the neurons express the CSF1 as an integral membrane protein on their surface, and the microglia interact directly with the surface-bound CSF1. Neurons also express the other ligand for the CSF1 receptor, interleukin-34. So there's a very intimate relationship between neurons and microglia in the brain.
Thank you. So this concludes the questions we can handle while we're live, but as I mentioned before, others will be answered by email. Finally, thank you again to you, Professor David Hume, for your excellent talk on CSF1 and macrophage biology. And thank you to your audience, all of you out there. Thank you for listening in, and goodbye.
Goodbye.